or concerns you may have. The setup is a bit weird, so I'm going to apologize in advance while I try to figure out how to best sort of deliver this lecture. I think. Let's see. All right. Let's give this a try. Okay, so before we get started, I'm guessing all of you come from a molecular/medicine background. Everyone here has heard the term genome editing before, yes? Okay, so what do you think it should be used for, or do you think I actually don't like it at all? If we have no need for genome editing? Anyone?
Like genome editing, like just changing between like long-term things and for better thing, or like for finding treatable thing, or like the disease like coming with the genome disorder, and also like secondly like for finding new therapy and new way of treating the disease.
Okay, cool. So does anyone agree with Sana, or do you have any different opinion? So her answer, if you couldn't hear it earlier, was ultimately she thinks that it should be used to focus on genetic diseases or as a therapy for diseases as a whole. Any other opinions?
I think genome editing should make food taste better.
To make food taste better. Okay, awesome. So those are two areas that actually anyone's actually looking at making food taste. You wanted to add something?
No, like but she's right. Like the two main things, like we are as a human focus, like keeping ourselves from disease and having food.
And having food. So I think a lot of people are focusing on food security. Sorry, I'm just gonna talk a little bit about it. So people are focusing on food security, but a lot of the effort actually not really focused so much on taste. But it's actually sort of focused on trying to make some of plants grow faster, or be able to be drought resistant, or really to be able to adapt to different types of environment. But yes, hopefully someday there will be people who make food taste better. Anyone else from this side of the class? Did you want to add something?
No.
No. Okay. Anisha, since you decided to join the class, I'm also just gonna hear your opinion, if you have any.
So,
Besides what we've already said. Okay, and three of you? No, that's okay. Hopefully you guys will feel more comfortable to talk as the class goes on. All right. So a couple of years ago, this is probably the most controversial point of anything thus far. So a couple of years ago, there was actually a researcher who modified babies, known as Lulu and Nana. People don't really know what has happened to Lulu and Nana today, but at that point, it was very controversial because he modified the baby so that they would be HIV resistant. And it was very controversial because you actually can prevent mother-to-child transmission with normal medication, and also HIV/AIDS is something that you also can. There are sort of other methods to prevent it. But let's just sort of hear what he has to say, and then I'll hear what you guys think about designer babies.
So beautiful, you go Chinese letter. This document change the future with human race and let the world know on the future. Bloodshed by still in the scientific community, with the theme we push the boundary, colonize.
It's very disturbing, but it's always a
This one?
Bill says he genetically edited human embryos for custom research for implantation, leading to the world's first works of genetically altered humans being brought around China with embryos designed to be resistant to HIV.
called CRISPR is found in labs around the world.
Give us the size, weight, and complexity of coding.
It's all researchers trying to do leaders in the field of gene editing have left the field
What? Ah.
So, questions about
Just pause it.
Yeah, sure.
这样知道了，也两个一起的应该是。
too beautiful, you could Chinese. This is the document which is the future of the human race and let the world know on YouTube. Too beautiful, you could Chinese there. This is the document which changed the future of the human race and let the world know on YouTube. Lo Zhenguai stunned the scientific community with the claim he pushed the boundary no one else could.
Oh, I think boss, it's not boss.
It's very disturbing, it's inappropriate.
Oh, this is huge.
He says he genetically edited human embryos, not just for research before implantation, leading to the world's first births of genetically altered humans, baby girls born in China from embryos designed to be resistant to HIV.
For this specific case, I feel proud actually.
The tool used by He called CRISPR is found in labs around the world.
It gives us the precise way of cutting the gene or putting a new piece of gene in.
It's often used by researchers trying to treat incurable diseases.
The method itself is very easy. You can use it everywhere you use it now.
Leaders in the field of gene editing have collectively agreed it's too early to implant edited embryos in humans because of the risks, the unknowns, and the ethical questions about altering humankind.
We're at a hinge of human history.
William Hurlett is a leading American bioethicist who teaches at Stanford University. In the months before the news broke, Dr. He consulted with him.
Every time we met together, we would talk about the seriousness of the issues and in a sort of stepwise way, what you had to do to make sure that it was done right. But when I heard that there were live-born children from it, I thought, oh my gosh, you just jumped ahead.
Hurlett knew nothing of the plans to implant edited human embryos. Dr. He studied at Stanford as a postdoctoral fellow where he worked with leading researchers. Hurlett describes him as smart, but perhaps too trusting of his own wisdom.
He's young, he's inexperienced, and he's from a small rural community in China.
This research has been shut down by Chinese authorities. It's also raised questions about whether there will be a rash of new regulations to stymie scientific development, or if scientists can regulate themselves.
We all want to be first, right? I mean, this is where you really feel that you make a huge difference and you get the recognition. But I think in this particular case, the outcry from the community is so huge that I think we'll slow things down.
He's work has already stoked fears about the future, what it could look like, how soon it could come, whether it includes designer babies, and if a tool found in labs around the world could one day make them. Alexander Field, CNN, Hong Kong.
Okay, so what do people think about this? Because initially some of you said, well, I think genome editing should be used for diseases. So do people think what he did was okay? If yes, why? If no, why not? HIV is a disease. Okay, I'm gonna pick someone sorry, what? It's complicated. Okay, while you think about how to uncomplicate it, so what do you think?
Effects.
You have a
Unpredictable effects.
unpredicted events. Okay, so then you might not know if there are any potential sort of side effects. Okay, right, what do you think?
Development something to the treatment of the cancer. Using the genomic analysis.
About, sorry?
Like genomic analysis.
So do you think that what the researcher did was okay?
Yeah.
Yes. Okay, all right. Gao, what about you?
是I think this research sorry
Oh, that's okay. Take your time.
勤奋怎么说？
Okay, well, Gao tries to figure out what he wants to say. Which one?
I think it's unethical because as far as I know, he didn't give proper informed consent to the participants and it was against the law. Okay, alright, and you? Basically playing ourselves. You never know what people are wrong. So simple.
Okay, alright. So it looks like most people. It's a no from most people. Gao, do you want to add on?
because it violates the human rights of the 10 babies.
Ah, okay. That's a very interesting point because it violates the rights of the babies. Because babies are too young to really sort of have a say. So all these are important things. Everything that you raise are all also important things to think about. All of you being molecular students will probably be involved in the development of something or other, really taking all those things into consideration. So for anyone who is curious, you can click, so all the slides are uploaded onto eLearning. You can click on the links and those, the information, you can actually read more about sort of the experimental details on how they created the babies using genome editing, as well as the regulatory framework. So as one of you pointed out earlier, there was a Nixon law in China, not only in China, but globally as well. So really what sort of framework do we have for genome editing? All right, so in the next 2 hours or so, 3 hours or so, we'll talk a little bit about the technicalities of how would you actually work with CRISPR. So the first part will really go over different CRISPR-Cas systems. A lot of people are probably familiar with CRISPR-Cas9, but there's tons of other CRISPR systems out there. So what type of systems do you have and what can you modify? How would you actually plan a CRISPR experiment? So this is something that you might want to do. You decided no one has made food here. I want to go ahead and do it. So how do you actually plan for the experiment? How do you validate CRISPR efficiency? This is also, it touches a little bit on some of the points that some of you raised, where you talked about adverse events, unpredictable events. So how can you actually sort of check whether it actually does what you want it to do? And finally, the implications of CRISPR. And this is really sort of tied to, again, some of the points that some of you raised about human rights, whether it's ethical or not. All right. So I think everyone is probably familiar with this. Genome editing refers to the ability to modify or edit the genome. I do say DNA over there, but genome editing today is a bit advanced that you can actually edit RNA as well. So you can insert, which means if you want to introduce a new gene into any organism, you can do that. So you can insert any sequence that you want. You can replace, so this is typically done for mutations. So if you know that there is a mutation that puts someone at risk for a particular disease, you can just go ahead and change that sequence. Or you can also remove something. So again, if you know that someone is, say, overexpressing something or someone expresses something, which again puts them at risk for a disease, you can actually also go ahead and modify it. In layman's terms, genome editing is often referred to as molecular scissors because of the mechanism of how genome editing works. All right, so really sort of taking a walk back to the history of genome editing. Genome editing actually covers these 4 main tools, even though what probably most of us are familiar with is CRISPR at the bottom. But before that, really sort of back in the 80s, I think we had actually meganuclease forming endonucleases. No one really uses this today. And then came zinc fingers, which is at the bottom. Actually, a lot of people in the field of agriculture and actually even livestock still actually use zinc fingers. And then came tolerance, which was actually really a lot easier to design and develop, but it was actually really difficult to deliver. I'll talk about why that's significant even a bit later on. And then CRISPR-Cas system, which is really sort of the golden child of all genome editing tools. If you look at the similarity between all those four systems, you can sort of see that there's this like two scissors sort of on the top strand and the bottom strand of the DNA. What that means is there's always a DNA cleavage component in the genome editing tool, and then you'll see sort of that orange, with that's spoke one, and then at the bottom there it's like a Cas system, but there's also a DNA binding domain portion in the genome editing tools. So if you have those two together with your DNA binding domain, that means you can usually develop your genome editing tool to recognize any DNA site. And with the data domain, it creates the cut. And I'll talk about why this is important later as well. Questions so far? Yeah, everyone's okay? All right. So the function of genome editing has really expanded quite a bit today. Sort of the old conventional function of genome editing was always gene editing or gene knockout. That's what you typically hear. You just want to knock out a gene. But today you can also use it to transcriptionally repress or activate a gene. So what that means is if you just want to knock down as opposed to knock out a gene, so you just want to silence a gene for a little bit, you can actually do that. If you want to overexpress a gene or just express a gene, you can also do that. You can also make epigenetic modifications. So with a lot of systems as well, sometimes there might be methylation or you want to de-escalate something. You can also use genome editing for that. People also use it for large-scale genetic screening. So in Malaysia, there is a group that uses that to try to identify what sort of works for different types of cancers. So Cancer Research Malaysia does that. Generation of animal models, genomic imaging, and lineage tracing. We'll go over what different CRISPR systems can be used for all of that shortly. Right, so even though this class focuses on molecular medicine, I really just wanted to give this really broad overview of how and where CRISPR can be applied. So the four main areas are therapeutics, biological research, agricultural biotech, and industrial biotech. If anyone's curious, in Malaysia, actually, people are working on all four. If you are sort of curious, if it's something that you want to pursue, but you're wondering where do people typically fund their research, there's a lot of work that goes, a lot of funding that goes into therapeutics, so that's gene and cell therapies, obviously to try to prevent or cure a particular disease. For biological research, that's really sort of your fundamental biology research. Agriculture, biotech, you can imagine with all the different types of crops, especially that you want to modify. And in industrial biotech, if you want to change any therapeutic bioproduction, so really overexpressing certain enzymes or microbial filtration. Questions so far? No. Alright, so this is a bit of an older table, but really just to give you an idea of even really sort of going back to the zinc finger days, there were lots of different genes that people have successfully modified. So if you look at some of the genes, a lot of that are disease related, and it's also been done in a lot of different organisms as well. So you see humans, zebrafish, rats, hamster, and so on. A lot of people have also tried introducing a lot of genes as well. Again, a lot of this is in the context of diseases. So you see things like OSA26, batch FA, and gene correction as well. Again, this is a bit of an older table from sort of 10 years ago, but you really sort of see, especially in the yellow section. So this was meganucleases, which is where a lot of the initial genome editing work started. It was a bit challenging to work with meganucleases, so you see that people really only worked in yeast, primate cells, and human cells for that. And then when zinc fingers came along, it was a bit easier to work with, so you really sort of see across the board all types of animals, humans, and plants. And then similarly for talents, with CRISPR, in the next slide, I'll show you how the field has grown. Okay. So the history of CRISPR started in 2012. There were two groups that sort of concurrently worked on CRISPR. There was a group from the University of California, and there's another group from Harvard/MIT that worked on it. And in the short span of 3 years, you can see, hopefully you guys can see this from the back as well, they were actually able to edit animals, plants, human, mouse cells, zebrafish, bacteria, monkeys, plants. They managed to send in a patent application, they did a gene drive, and they also used CRISPR to edit human embryo. Very, very fast, all in the span of just 3 years. It was also named Breakthrough of the Year, and there's also a Nobel Prize that was won because of this. And this really sort of goes back even from the St. Finger days. So Nature actually called St. Finger Nucleus Method of the Year in 2011, while Science called CRISPR Breakthrough of the Year in 2016. Alright, so why the big deal? Why is CRISPR so much more special in comparison to a lot of other therapies or a lot of other systems we have there? So in the next couple of slides, it's really just going over to give you guys an overview of what CRISPR is. So it was naturally, or it's found naturally in bacteria, and it actually functions as an adaptive immune system. So what this is usually you can have phages that infect bacteria, and usually when you have this phage that infects the bacteria, it releases its DNA into the bacteria. In very simple terms, once the DNA is actually introduced, the bacteria is then capable of recognizing, hey, this is not my own DNA. And it sort of chops it up into really small pieces so that in the future, when it sees this same genomic information again, it knows, okay, we need to get rid of this. And how it's typically done is it cleaves that DNA. Once you get cleavage, that sort of disrupts the entire genomic sequence, and that's the end of whatever genomic DNA that was introduced. So it's a really cool way of that exists in the bacteria where it can actually prevent itself against all sorts of infections. Even in this sort of natural system, you do sort of see the core components that enables bacteria to protect itself, include all those things at the bottom here, which is your Cas protein, pre-CRRNA, and CRRNA, which stands for CRRNA. Questions so far? No? Everyone can follow, okay? Okay, you want to leave? Is it that bad that you have to leave? Do you want me to stop? Okay, I'm just gonna continue. Okay, all right. Hopefully, Rad can follow along. Otherwise, maybe one of you can help him when he gets back if he doesn't understand. All right. So, really sort of thinking back about the previous very complicated looking system. Obviously, that would be quite hard to use in lab. So people then sort of made it a lot easier. They simplified the entire system. So what you see at the top, that's actually sort of the first generation of CRISPR. So it actually still did sort of have that CRRNA that I pointed out, and then it had a tracer RNA, and then it had all these other things called a spacer and PEM, and then of course Cas9 protein itself. But then people simplified it even more, and you have what is at the bottom today, which is really your Cas9 and your guide RNA, which consists of both your CRRNA and your tracer RNA system. So again, just to recap or summarize this entire slide, when people talk about CRISPR-Cas9 or CRISPR-Cas system today, it consists of two components. So one is your guide RNA. So your guide RNA is the part or the portion that is able to recognize and sequence. And the second part is Cas9, which is what enables cleavage to happen once it binds to the site of interest. Great. So again, this is the CRISPR-Cas system. So much simpler than what we saw earlier. You have your guide RNA and your Cas9. And we'll talk a little bit about the PEM site as well. So the PEM site is a little bit like the stop. You need a PEM site before your guide RNA is able to bind. The PEM site is usually specific depending on the CRISPR-Cas system that you're dealing with. For the ones that people commonly use, it's the sequence NGG, which means anything that's sort of upstream from that is a potential guide RNA site. So if you don't have NGG in your organism of interest, that actually means you can't develop your CRISPR-Cas9, or you can't develop a CRISPR-Cas9 to the organism. That was very complex and complicated. Can everyone follow that? Or do you want to read it? The time again. Okay, and then use the board. Yes. Okay. Okay, so when you talk about you have your guide RNA plus your Cas9. Your guide RNA is what binds your DNA. Everyone okay with this? Yes. Okay. So even though we like to say that CRISPR-Cas9 can bind to any DNA sequence, what you actually need is this thing called a campsite. Without this campsite, you actually can't get your guide RNA to bind. In a lot of the CRISPR systems, the campsite is NGG. So N is an A, a C, a G, or a T. Okay. So if you have this sequence somewhere in your genome, that means anything that's upstream of it, that's your possible guide RNA set. Okay. So if you don't have this, that means you can't find your CRISPR to get site on the genome. This side is okay. Down, okay. The back, okay. All right. So CRISPR requirements are also pretty simple. Again, you just need your campsite. So that's the NGG site over there, and then anything that's unique. So earlier we talked about things like unpredictable side effects. So a lot of times these unpredictable side effects comes from what is known as an off target. So an off target is, say what you want to do is you want to knock out, are you okay? Yes, okay. Say what you want to do is knock out gene A, but you end up knocking out gene B and C. Is that a good or a bad thing?
Sometimes it's good.
Sometimes it's good or it's bad. Okay, we'll come back to that. This side. So again, I'll repeat the question. What you wanted to do is say you knock out gene A, but you end up knocking out gene B and C. So what do you think?
It's fine.
You think it's bad? Why?
It's not happening. It's not gene B.
Okay, because your target is not gene B.
Yeah, say that why do we want to go B and C?
Okay, Sana, but you said it might be good. Why?
It's not good.
Let me rephrase. What you have done is you develop something that can successfully knock out COVID.
So many were rather than using like the spike cases. So people are worried by reach Malaysia. So someone in this class was like, I'm going to develop CRISPR to knock out COVID. Say you knocked out what is known as a spike, or what you wanted to knock out was a spike protein. But you ended up knocking out two different genes in the virus.
The virus turns up highly virus.
You could also get rid of the virus.
Yeah.
So, but you guys are right. A lot of times it is usually not so good because you usually don't want it to be specific to your gene of interest. All right. So again, really sort of talking about the mechanism still. Your DNA binds to your packet sequence, your Cas9 cuts. What happens after that is usually your cells don't like when there's any broken DNA in your cells. So usually you will attempt to repair the broken part, organ cleavage. Is everyone here familiar with NHPJ? That you learn maybe in your degree? Some people say
And then homologous entry.
yeah, have you guys learned about this before? Who hasn't learned about this? I'm okay to talk about it if you haven't. Okay, so I think there might be one or two of you who are not with them. So your cell can repair itself through two methods. So either non-homologous end joining, which is the more common method of repairing your cells. Non-homologous end joining is actually a very sloppy repair mechanism. So when there's a cut, what it does is it randomly inserts or deletes sequences. So when you have that in the next slide, I'll go over it in detail. What actually ends up happening is if you imagine that there are just like suddenly you have one extra nucleotide in your sequence that causes separation. Is that good or bad? What is separation? Is that a question? Yes, correct. So yes, if you do have like a single base pair insertion, usually you end up with something completely different. So it might disrupt the function of the entire protein. Was everyone able to follow the explanation? Yes, okay. So non-homologous end joining is typically what enables a gene to be knocked out. So that's how the knockout function of CRISPR usually works. Homology-directed repair is a bit more complicated, so I'll actually use images to explain that. So again, this is how non-homologous end joining repair works. So you have your cleavage over here. You get random insertions or deletions. So regardless of whether it's a 1 base pair insertion, a 2 base pair deletion, a 3, a 4 base pair insertion or deletion, it ends up changing everything downstream. So you end up with a very different protein product than what you initially expected to get. So it's not only that you end up with a different protein product, sometimes you may also get a nonsense codon. So you get a stop codon happening very early on, so you may not even get a full protein. So again, that results in a loss of function or a knockout of your gene of interest. Everyone okay? So, now okay? All right, so HER on the other hand doesn't happen as frequently, but people actually use this concept to knock in, to introduce a new gene of interest. So whenever you get a cleavage, you can develop or you can also deliver a donor template. The donor template usually contains the gene of interest that you want to introduce. And because of the homologies between the donor template and the original sequence, you can actually introduce a new gene. Some things to sort of point out, your repair template can be used to edit as well, and it has to be homologous to the original sequence of interest. Okay, so that's the basics of CRISPR. So over the next probably about 15 slides or so, it's really about the different types of CRISPR systems that are there. Alright, so number one is the multiplex system. So the multiplex system is saying what you're doing is you are studying something in the cancer pathway and you realize that if you knock out 7 different genes, you can cure cancer, hooray! So a multiplex system is really the ability to knock out up to 7 different genes concurrently. The other system is called GECKO, so this is related to the genome widescreen that I mentioned earlier. The GECKO system is really a full CRISPR library. So people have actually developed CRISPR library where every single gene of humans and of rats have been actually, you can knock out every single gene in humans or rats. So this is actually quite useful if you're trying to identify a particular phenotype or you're trying to study different drugs. Especially in the context of drug resistance. Anti-CRISPR, so something that sort of comes up quite often is some people say, okay, if I'm going to use CRISPR as a therapy, maybe I don't want it to be hanging out in the person's body forever. Because, and a lot of you mentioned this, you don't know what the effects might be. So even though this hasn't been developed for humans yet, at least in plants, people have developed CRISPR so that it is an inducible system, which means you can control when CRISPR is expressed or not, actually with the ability of genes that are sensitive to light. So that's actually quite easy, right? Especially if the plant is growing in a greenhouse, you turn on the light, CRISPR's expressed, you turn on, CRISPR's not expressed. Unfortunately, humans don't work that way. If you turn off and CRISPR doesn't express, that might be kind of weird. All right, This is another alternative. So I mentioned CRISPR-Cas9 quite a bit. This is a certain variation of CRISPR-Cas9. It's known as SA-Cas9. So if anyone is curious, the first in-human trial that was actually done to actually treat cure HIV was actually done with SA-Cas9. The results just came out. It was not successful, unfortunately, but SA-Cas9 was what they used. And they used it to deliver into humans because it's a little bit smaller than the conventional CRISPR-Cas9 that people typically use in labs and plants and whatnot. And it can also be packaged into AAV. I'll talk about this a bit later as well. And what's also interesting about this is it targets certain regions. Alright, the next system is known as a nickase system. So with a lot of the systems that we saw earlier, they all have double-stranded breaks. But in the early days as well, people thought, okay, if what ends up happening is, again, you want to target gene A, but it might target gene B or might knock out gene C. Is there any way we can prevent that from happening? So someone thought, okay, well, if we develop it so that it only cuts one strand as opposed to two strands, might the possibility of off targets be reduced. So this is what the nickase system is. Okay, everyone okay so far? You guys need a break? I think some of you yawning. Is it because it's after lunchtime? Maybe, yes, kind of boring. Maybe that's okay. If the answer is yes, you don't have to let me know. All right, so other systems that are out there is the reproduction and activation system. So this is known as CRISPR-I or CRISPR-A. So the repression system, you have your normal CRISPR system, but you have an additional inhibitor or transcriptional repressor attached to it, so you can actually downregulate the expression of certain genes. Again, this might be useful about something like cancer, where you actually have a lot of genes that are overexpressed. You also have CRISPR-A, which is to activate certain genes. So, for example, if someone, for some reason, does not have high levels, say, a tumor suppressor gene like P53, then you can actually go in and increase the amount of P53. So, all of this actually, a lot of it is actually the profile types of diseases, epigenetic modification. It's really just a summary table between a few of the different CRISPR systems that we have learned. So again, with a lot of the modifications for DNA, that's the Cas9 system. We talked a little bit about Cas12a. Again, that's a little bit smaller, that has actually been tested in different sort of human trials. And then 13a, which is for RNA and sort of naturally found in bacteria. We do see double-stranded breaks with both Cas9 and Cas12a, and with the thing, it's RNA cleavage. Right, another video.
Gene editing tool could fix harmful genetic mutations. Science Institute of Technology have developed a new method of editing DNA called prime editing, which combines CRISPR-Cas9 with a reverse transcriptase enzyme. According to a study published in the journal Nature, the CRISPR-Cas9 is used to nick one DNA strand, while the enzyme generates a new DNA strand, which is inserted into the original DNA strand. Another reverse transcriptase enzyme then guides the CRISPR-Cas9 to nick the other DNA strand. The strand is repaired as the enzyme replicates gene edits over from the new DNA strand, producing a fully editing DNA strand. According to a Harvard News release, prime editing can also be used to replace one DNA letter with another, and could correct up to 89% of genetic mutations. In the study, researchers explained that they used prime editing to make more than 175 edits in human cells in a laboratory, and successfully corrected sickle cell anemia and Tay-Sachs disease. According to the Harvard News release, CRISPR-Cas9, if used by itself, isn't as precise as prime editing, and could lead to mismatched DNA strands that either include extra DNA code or are missing a certain genetic code. The authors of the study concluded by saying they will investigate the effects of prime editing on human cells and will continue to test prime editing on cells.
Okay, so what's different from scientific editing?
Okay, what's different from prime? Very funky video. What's different from prime editing and APS? Is if you recall the details from KTD, for example, that converts specifically A to G, and C to U. But if you say the mutation is an A, but say the polymorphism on the SNP that doesn't put someone at risk is a C, so any of those earlier systems wouldn't work, and you would need with something like prime editing. So prime editing really gives you a bit more flexibility. Right, we are done with the first part out of 4 parts. Any questions from anyone so far? No? Everything was understandable?
I question like after like look at my gene that was on the thing. The DNA repair system will come and insert the gene complete like this replication, and then maybe this is like the idea of CRISPR-Cas9.
So the idea of CRISPR-Cas9 is that so once you have a cleavage, usually it's required by an which inserts or random insertions or deletions. So usually you can't control that. So once that you can yeah, so it because it's a very sloppy pattern, which means you don't embed anything from a core base pair insertion. But if we look at the different molecules, so if you actually were to do something like NGS, so you will see one molecule might have 3 base pair insertion, another molecule will have a 6 base pair insertion, another molecule will have a 50 base pair insertion. So it's quite intense. But regardless, usually all of those insert or emails would usually result in a frame shift. So, and because of that frame shift, then the function of your gene that you targeted is not targeted.
We disrupted the function for that gene. So what about like inserting the evolution for that?
Okay.
This is like this is how it's working.
How will it work? Okay. So, okay, I will, maybe I'll use the board. All right, so I don't know if everyone had, you guys are okay? You guys had a question? Yes, okay, all right. So I'll come back to you guys because it looks like you guys have a very interesting discussion going on here. So, so the question was really sort of after having gone through all the different sort of systems, how would I be able to summarize how CRISPR works or what does it do? So the first part is really once you have a cleavage, it's repaired by non-homologous end joining, which randomly puts in any number of bases as an insertion or deletion. Which means if you do something like NGS, you would see one molecule might have a 1 base pair insertion, another molecule might have a 3 base pair insertion, another molecule might have a 5 base pair deletion. So it could be any of the above. You can't actually control that. But ultimately, because you sort of have all these really random insertions or deletions at your cut site, that results in a okay, so that's another decision. But that's why I'm not going to there you have your okay. So you might sort of say, all right, for or somebody might say, okay, I think I feel like I it's very, very rare that your cell okay, what is your concern? I shall try to answer it with hyper. You okay? Really? Okay. You guys are okay? No. Okay. Down, okay? All right. So hopefully everyone understands. Yes.
So you're saying when the gut is made and let's say the strength rate is
Yes.
even the inside.
So not any yeah, so identify. So you do two steps. But not CRISPR-Cas9. CRISPR-Cas9 all it is only pair is DNA. No virus. So assuming this has a number have a mutation in exon 1, a mutation in exon 2, a mutation in exon 3, which is not everything else. Sort of say, okay, I'm just going to target it. Well, maybe with this one. Same thing. Yeah. So what you do is you actually first, so that you will find here really close, and then you will actually so the template will contain your new gene, and then it will be homologous to your original. So when it's homologous to your original, usually you have that's why you get it from homologous directly. So the first one is what your So if you say I want to prevent cancer, great. You have to know your cell line and cell. Should also select your team. We ask the right question. Actually, a lot of it is incorporated. So Well, in this recap, you can mark out as a press or activate the gene. You also can choose it so that it is something that can be introduced through different virus systems. You can also introduce it as RNA or as protein. All right. If people want to follow along with this, so if you want to follow along with this, so your demo tomorrow will have to look like this. But okay, so for anyone who has gotten to that part of the website, okay, so you will see on the website. of biology. If you click something on LinkedIn and everything, you will see there are a lot of just to show you that it can be quite diverse depending on what. Also, all right, then I'm gonna make sure I do something GFP because I have to visualize it easily. Or if you're a lab that works with antiviral vectors, then you can also choose something like this because you know that you have the capacity to work. Yeah. Antivirus work, I think I find it other types of things. So I think the great diagram. top so assuming CRISPR, so you just pick SPCAT and you input your features. It will give you a readout. Sure, guide RNAs, where is it located in the gene? What are the potential mismatches? Short is just about 300 bases, but in the span of about 300 bases, you actually see that there are possibly 25 different guide RNAs that you can design in that. So that's actually quite useful because, yeah, even if you're looking at you have probably 5 different possible guide RNAs. This website I really like as well because it already tells you whether your guide RNA is low efficacy, medium efficacy, or high efficacy. So in this case, it's probably a medium efficacy, which means that it might work okay. Maybe not the best guide RNA that you can find. So maybe the other information at the bottom here is if there are possible off targets. So if your guide RNA could somewhere else, what could it bind to? So it tells you that it could possibly bind to this genes CRMP1, ZPLB1, and so on. And it also tells you whether those bind to the exonic, intronic, or intergenic region. We'll go over all this in detail tomorrow at our demo. So very similar. So hopefully by it's also quite similar. So this case, you'll see what your potential guide RNAs are. It gives you a predicted efficiency. In this case, you see some with very low efficiencies, like 20 at the bottom here. If we see something like this,
Seems like very like is it like final design? It's like there's some rules with like the 14 to 16% content or this and that. Is there any rules for it's like
okay.
啊，我，听到了，操，哔哩哔哩，哔哩哔哩，no, yeah, yeah.
蛋白是叫做氢蛋，在这里，这里就是氢蛋，它就是因为双链点切。就是在它输入的哪段位置点切呢？谁来告诉他现在需要剪切哪里？这时候就需要一个 FgRNA。这个 FgRNA 和我们通常实验中所用到的 FgRNA 是一样的。我们这部分的 FgRNA 包括两部分，它等于 tRNA 对 RNA 和 tRNA 是两部分不同的。而在这个图中呢，这个 tRNA 它是个蓝色的部分，而这个 cRNA 呢，它是这个黄色的部分。它们两个在末端。谢谢。
第二个。
So, okay.
我就可以找在别区 overlap， 对，因为他们有一个重叠区 overlap， 就可以这个部配对结合在一起，然后这样的话 FDRA 到这半部对外元素性的 DNA 进行剪切，这就是一个最简单的 CRISPR 实现 DNA 剪切的一个功能。那么接下来就要问了，CRISPR 蛋白是哪来的？往上看，CRISPR 蛋白这是一个CRISPR 蛋白，这是一个CRISPR 蛋白，而这边呢，除了有一个CRISPR 蛋白之外，还有一个 CRISPR、CF 三个蛋白。在这里我们需要知道的是 ，CRISPR 和 CF 它们这几个生物体被当做 K4 open 这方面，它们几个总体在一起能够产生一个有功能性的病体。
And what's good about it? way of explaining to cells. It's really easy to use, but the downside of it is it's usually shortened, and it's usually not for long-term users. So again, this is more used in labs than anything else really. Right, so just a very quick comparison of the common techniques. So between lipid-based reagents, electroporation, microinjection, and virus, if you talk about what is sort of more efficient, definitely virus and microinjection are the ones that typically have higher efficiency. The principle, they are all pretty different. So liquefaction uses like a lipid, which is complex with your CRISPR. Electroporation is really sampling the cells, but you do need a specialized equipment for that. Microinjection, you also need a specialized equipment since you're actually injecting it into the nucleus. And with virus, you actually need the capacity, all the facilities to actually produce viral particles. With limitations, again, with virus, you need sort of a very safe and secure lab. With microinjection and electroporation, you need specialized equipment. With cell types, with electroporation and virus, they offer flexibility. So that's why a lot of people use electroporation for hard to transfect cells and virus in general for gene therapy. Okay, these are some of the other methods. CPP, I think someone or you guys were nodding when I asked them this is all kind of definitely inform. This is really sort of complexing it with your CRISPR and then delivering it through endocytosis. It works pretty well. There's also nanocapsules or nanoparticles. I think there's also quite a lot of work on this being done in INFORM. There's lots of different types of nanoparticles out there, so I'm not going to go over this too much. Nanoglates, this is viral based, but this has only been done in certain cell types. It's based on mouse leukemia virus. But what's sort of different about it in comparison to sort of lentivirus and AAV and adenovirus is it's non-infectious and non-replicative. So if you need a system that is sort of able to sustain itself in the body, because imagine you have so many cells and you need something that's not replicate along, this may not necessarily be the best option. Right, this is for bacteria only. It's interspecies conjugated transfer, very fancy name, but it basically works the same way as conjugation, which I think you guys would have all probably learned when you were doing your degree. All right, we are almost at the end of the second part. So assuming you have picked up that array, you picked up the system, you cloned it, you have successfully delivered it to your cell of interest. So now you want to know whether it did work or not. Right, so in this case, you can validate it through a few different ways. All right, so I would say if you don't have any of the options, these are some of the main ways to check whether the cells have been modified. So one is known as the mismatch cleavage assay. This is a bit complicated, so I'll get to the slide and talk a little bit more about it. Restriction digest, I'll also get to the slide and talk about it. Certain things are quite easy, for example, PCR amplification. So say what you're doing is you're inserting a gene, which is 1 kb in size. So it's very easy. If you just PCR up the whole region, just check is that extra 1 kb there. Cellular sequencing is the same, especially you can use this whether you inserted something, you knocked out something, or you did a base edit for something. They will be able to tell you all of that information. Questions? No. Okay. Right, so we will go into the nitty-gritty. So you will see this. So this is just in one small region, but this is yeah, so that we all right. The Right. Increase the energy production. So this actually has picking the distance.
Channel. 是事情。
Only hear the Lulu and Nana.
In my country, there's one gene because some people in south of my country, there's so many people. There's so many people in the original gene in my country. But you found some disease from because all people now found genome disease in the
Okay, so maybe it's not so much yet.
now found the search project with the GBank, with the British Council.
Ah, okay.
Yeah, about of the research about of the original gene in this area.
Oh,
Yeah.
interesting.
Yeah, found some bone
Yeah.
in this the was great, the summary.
Mm.
Found the sun bone is old bone.
Mm.
Found the sun analysis the bone splitting the DNA.
Mm.
Comparison between DNA between the people find in this area and all
Different.
no same. Same pricing.
Okay, so high similarity. Okay, all right. So if there's high similarity of sort of genetic makeup in a particular area, probably people may not be okay with this. Maybe, yes? Don't know. Okay, all right. Interesting. All right. Thank you so much everyone for sharing, your input and insights. Because it's always interesting to sort of hear how people view all of this. All right. If anyone's curious, you can actually also just Google sort of clinical trials and all that, and there are actually lots of websites now where they give you lots of details of all the ongoing clinical trials, what phase they're at, how many people they've recruited, when they're expected to finish. So the one that I was talking about, where the results were maybe not up to par, they were actually also going to try with a higher dose. So that's probably what is happening next. And they're actually also going to monitor people for 15 years, which is a very long time. But it's also good knowledge because currently we don't know. So if after 15 years the patient is great, there's no other sort of adverse effect, that would be good for the world of genome editing. Right, so these are some of the examples that are out there. So as you can see, a lot of it does focus on sickle cell and beta thalassemia, this sort of more. And but there are also some hereditary diseases that people are looking at, and cardiovascular diseases that people are looking at as well. Again, if you want to know more, you can actually just Google up the different trial names, and you can find out more of them. All right, technical barriers. So one of the main reasons why some people are hesitant to take up genome editing as a gene therapy is the technical barriers itself. So I like one of the points that I think who sort of brought up earlier. You don't know what could possibly so a lot of times what we look at possible to get permission from the embryo for treatments. This really has become a big question, especially after Lulu and Nana. A lot of concerns have also been about accessibility. So currently, it's roughly estimated to cost about $5 million per patient, which is a lot of money. There are certain sort of agencies and institutes like IGI where they're sort of trying to come up with alternatives to enable it to be cheaper. There are also sort of other organizations where they are sort of training different countries to be able to develop CRISPR in their own country and mostly to manufacture CRISPR. Manufacturing is what's really difficult. But if that's not possible, is that okay? So something that we talked about, so I think ING also did we were to tell something that sort of came up was someone asked, what if the ultimate gene therapy costs $5 million, but a somewhat okay gene therapy costs $100,000, and that's something that more countries can afford? Would people be okay by saying, okay, the $5 million one can go to richer countries, and the $100,000 one, which works only sort of okay, go to other countries? Do you think that's okay or not? Who says okay? I thought someone was going to raise their hand. Anyone thinks that's okay? No. So I think, you want to add something?
No, I'm saying like this method of trials already like it's a risky treatment, so even if you feel like for a riskier so it's not.
Yeah, correct. So that's a good point. And that was the point that a lot of people raised as well. So we call this a target product profiling. So when we were sort of developing what we perceive as ideal and useful for HIV gene therapy. So ideal might be less than 1% adverse effects. Minimum might be less than 30%, which is actually quite large, right? That means out of every 100 people who take this therapy, in an ideal situation, maybe only one might have any side effect. But in, like, for the one that's not optimal, maybe 30 people out of 100 with side effects. So it's quite interesting. So yeah, that was sort of a big thing. And then this I sort of asked you guys earlier, is it okay if people genome edit for aesthetic purposes? So what if someone says, I want to be taller, it's up to me what I want to use my money on. Or someone is like, well, I want to be faster, it's up to me. I can make myself run faster. So, and that's something that people also starting to think about, because if people use it, especially in sports, there's no way to be able to detect that. So a new form of doping. But then finally, should scientists be able to edit germline cells? I think a lot of you, all of you, said. So this, I think this is based on Singapore's results. What obstacle do people believe is the highest priority for gene therapy? So most people actually answered risk associated with the actual therapy itself. The second most highest reason was ethical and religious issues, and then came how easy is it to use it and economical issues. Right. So this is based on the US actually. For any questions from me? No. Okay. I think we're almost at the last few slides. So morality of human gene editing. So this was really sort of a survey. What do people think about gene editing to give healthy babies a reduced risk of serious disease? So it's quite interesting because in the US, sort of among all the people that they surveyed, most people actually say not sure. Those who considered themselves highly religious, almost 85% said it's morally unacceptable or not sure. Those who considered themselves moderately religious, a lot of people still said unsure. And then those who considered themselves non-religious, they were the ones who were in the highest group of people who thought that it was okay. And then in the same sort of group of the acceptability of gene editing research, so they also asked them, so if you were to edit healthy babies again, would it make a difference if there are different amount of risk? So if I tell you, I can edit the baby will be fine, versus I can edit the baby, but I'm not sure if the baby will be okay or not. So what do people say to that? So if people said, if the question was if the effects were limited and could not be passed on, which means that it can't go from parent to child and so on, a lot of people said it made no difference because you are trying to edit a baby, right? Why would you think about sort of the next generation? If it required testing on human embryos, most people were actually not okay with it. So in general, everywhere in the world, a lot of people are just not okay with human embryo editing or testing and germline editing. If we change the genetic makeup of the whole world, the whole population, most people are also not okay with it. That would be kind of scary. All right, I think this is the last almost slides to go. But did anyone want to add on to anything that we saw already or give your opinion? Okay, so then we'll just go to this. So public dialogue again. So in a lot of countries we are planning to do this. So with local communities, we are starting to talk to people, even if it's 5 years or 10 years ahead, because if there are anything that concerns the public, we have time to actually try to address those concerns before something is actually done. For example, if we want to sell genome-edited bananas in Malaysia, but people are not okay with it, and they tell us, oh, because I want it to be labeled in the supermarket, or I want it to be in a separate aisle in the supermarket. At least if we know about all this now, then we can actually do something to sort of make sure their concerns are addressed. Similarly, for medicine as well, if people sort of say, I'm okay with trying to cure sickle cell anemia because it's quite a that, but I'm not okay with antiviral methods. So okay, then we can use cellular-specific virus instead, or maybe we can use non-replicating virus instead. So we do quite a lot of this. And we also communicate to the public about the consequences of gene editing. So I think when we wanted to release the mosquitoes 13 years ago, there was a lot of sort of press conferences, and people with the team behind it tried to make sure the information was put out, and people could sort of write in at any number of time because mosquitoes can fly. So people were sort of concerned, what if the mosquito bites me? Like, would then the editing that was done on the mosquito, would I actually have it as well? So those were all very legit concerns. So these are things of what people sort of said. You can read more about this later, but some things I sort of wanted to point out is a lot of people are actually concerned about the genomic data. So this is quite interesting because if you are trying to modify something in your, say someone is at risk for a particular disease, and if you change something, it makes them less at risk. Some of the concerns were sort of, how do I know my insurance company won't ask me to do a genome sequencing of myself and then tell me then they're not going to cover for something? Which sounds very far-fetched, but it's true. We may not know if something like that happens. Other things to sort of point out about is some people say they want to know how would that affect their relatives. So for example, if we take patient A and patient A is cured, does that mean automatically anyone else in the family who has that would also sort of be eligible for the same type of study or the same type of editing? What else did I want to talk about? Taxation, so some of it is related to money. So some people sort of say, okay, we're paying X amount of taxes, so doesn't that mean that everyone has to sort of have equal opportunity for that? And then policymakers, but this is again sort of related to public dialogue. So a lot of times people want to have representation. So for example, if you're trying to modify issue of someone with sickle cell anemia, they want to make sure that people with sickle cell anemia are actually part of the research from the beginning, which means they get to say things like, I want to make sure this is a one-time treatment, or I want to make sure that this is something that I can take as an oral sort of pill, or I want to make sure that I don't have to come back 10 times for a follow-up. So these are all things that we also take into consideration when doing genome editing work. All right, that's it. So the last part is really a discussion session. If people want another sort of quick 5-minute break? Yes? Okay, 5 minutes break. I'll extend the rules and the discussion. So the discussion session is graded. I think it's 5%.
This is the last class. The from the start of the research. I don't know, in patients like this, much requires
In foundation, we haven't seen that, but so I studied with us. So it was actually quite nice because we had sort of different centers, and what they made sort of all the training research group was properly great. We actually represented so they could ask a lot of questions. So usually at the beginning stages, if say I will show you what's equal to my cell function. So the questions would probably be along those thoughts. how will that translate to giving? So it's actually only at the closer it gets to some of the clinical trials is when people actually have more and more questions and they want to be more involved. But I think especially with Chinese, because they have a very, very large and strong advocacy group. So they're very vocal about running along. So they are actually considered in some ways a very huge success story because they drove the price, because of that. They drove the price of education from was $10,000 to now it's less than $100 just by saying you should do this.
Because when people are frustrated, usually will try to agree to any terms that we hear.
That's also difficult. So I think people are becoming more empathetic because of the internet. So even like sort of COVID itself, a lot of people had a lot of opinions about vaccines. So I think now what is probably something to address is more how people identify and distinguish what is real or real vaccine versus something that is maybe very theoretical. So when we were swimming a little bit on us most recently about the mosquitoes. So it's quite interesting because the countries that we wanted to really see first, all of them were saying, but we don't know if it works. So can you go really see the different countries first?
But the worst case scenario, what the worst case scenario?
But all of us must know that it's it. So like with Indonesia, so we would do it that we just of solving the solution. So for us, I think we want to solve their diet, but not all countries can. Which for us, we don't think about it because it's so common. Everyone should have gone to right? Even things like okay, trying to identify is something genuine or paranoid. For us, PCR, most labs, most hospitals all have like a thermocycler or PCR machine. But in countries that don't, so one country was like, but we need to send samples to a different country. So then all of us were like, that's true. We didn't think about that, right? So then it ends up becoming a lot more expensive and it's not doable for those countries. Okay. Are people ready to start again? Final few slides. All right, let's go. All right, so for today's discussion session, again, this is about 5% of your grades, I think. It's really just to see what you think about this special. I will also take into consideration all your comments throughout the class today. Thank you again for being so active and participating in the class. So recently, sorry for some of the
Yes. Yeah.
change the humor now.
Because the question this
yeah, it doesn't matter. So I want to ask curious to know what you guys think.
Because now the development, they use the gene treatment, because use the stem cell,
Yeah.
anything.
So you would use it on humans?
Yeah.
To make them resistant to TB?
Yeah.
Okay.
Now the vaccine, same the corona vaccine. It's not active now because with some years found the problem in any vaccine, any treatment, need development. Now can use the genetic treatment, good, but found some because can use the mutation in RNA.
Okay, all right. So I am going to summarize because it is a very interesting point. Rod's comment was, even if you sort of look at SARS-CoV-2 as an example, and really sort of see how it's changed or evolved over the years, we see that a lot of the vaccines that were developed earlier may not necessarily be as efficient now. Similarly, with TB, we actually do see a lot of multi-drug resistant TB, which actually makes it very, very hard to treat because first line and second line of drugs may not work anymore. So Ryan is like, yeah, why not? Why not do anything then? Because that might actually sort of prevent sort of future mutations in the microbe as well, and it might sort of solve TB once and for all. Thank you for that. Okay, anyone else? For this, everyone needs to talk because it's 5% of your marks.
For the same to we have the same idea as that, like because nowadays, because of the aging of the population, and also like the evolution of the immune system, also all the things infectious disease and one of them like trigger resistant one. So all the focus right now is about like immunomodulation of the immune system, and one of the options is using like CRISPR system for gene editing on immune cells.
Okay.
For example, like macrophages, they're from the first line, how to like use that adaptive system from that human being. So yeah, like using that gene editing, maybe next decade it became the only option for dealing with TB, because I think like most of the research about antibacterial activity about what TB is working.
Yeah.
So I think like approach because of immunology channel.
Okay, all right, thank you. Okay, so that was also a very interesting point. Sana's idea is to modify something related to the immune system, especially again, sort of given that a lot of drugs don't work and we don't find anything new, especially soon. You are going to have a really huge problem because as long as you don't treat it or control it, that means the number of infection is going to keep on increasing. Thank you. All right, who's next? Sure.
I disagree.
Okay.
To edit on human.
Okay.
First of all, genome editing is not perfect yet because the technology are not perfect. Especially to interact with immune system, it's very complex. Once you interact with the immune system, everything will become
haywire.
Because everything is so connected and complex. So one thing that is changed you might resist to TB, but you might be more susceptible to other bacteria. Okay, and then another thing is that actually TB is the coughing that you see the symptomatic symptoms a lot. But if you can actually, if you want to know, actually latent TB is more a problem now. There's 25% of the world population actually have latent TB, which you are asymptomatic with. So if I am asymptomatic with, why do I go for genome editing? I don't have symptoms, why do I need to genome edit myself? So that is, then the problem is, if I genome edit myself, if I have latent TB, then suddenly I become like other problems with the immune system, I don't know. So this is why I'm disagree. But I may agree to genome edit the TB.
Okay.
So that they are not so multi-drug resistant. They make the whole multi-drug resistant plasmid on one. Just whole thing just disappeared so that the TB becomes easier to treat. something like that. Yeah.
Okay, thank you. Also, very, very interesting perspective. Earlier mentioned latent TB is a larger problem. So that actually does raise a good point, right? If you don't know if you have it or not, and if you do attempt to genome edit the person, will it even work? Not necessarily, unless you sort of just try to test everyone for latent TB as well, and then only if there are no cases of latent TB or active infection, then you genome edit. But you can also say sort of in general, and both their points are sort of related. You never know if you change something about the immune system. How do you know that you're not putting yourself at risk for something else instead? So I do like the idea of trying to eliminate plasmid that causes the drug resistance somehow.
It's probably good. Probably you're gonna do with the TV, but it can never be with the same item that you think.
Like if you were to actually like design this in real life. So if you were targeting the
TV
itself, right?
Yeah.
Yeah. So that's
So this is another like perspective, but it's reading the item.
Yeah, so that's also a good point, right? Because sort of when you try to think about therapy or treatment, you also have to know how would you deliver it to someone. So if it's something like HIV where you know, okay, the person is infected with the virus, it's very clear, you just have to go to people who have been diagnosed. But in this case, you probably can't like just spring it and think, okay, that's it. People would inhale it and then that is the gene therapy. I mean, I'm sure someone would think of that because there are sort of different sort of treatments where it is sort of based on equating. People don't know this would work, but interesting. So anyone who's sort of thinking about it, lots of prospects and lots of areas to explore.
But I think like this is also this is one of the approaches that right now working on the CRISPR treatment, like the idea like delivering like the CRISPR inside the human body and work there, like because I think like still like they're working on the CRISPR and to the human treatment eventually it was like on that lab and then you transfer like
correct.
Yeah. So I think this is enough.
Yeah. So
they didn't found yes.
Yeah. So delivery is which is why it's easier to sort of kill certain viruses or certain diseases, especially if it's single gene. But something like TB is definitely complicated. All right.
You edit the ethical concern. You edit human ethical concern.
Yeah. Also true. Yeah. Good. That's actually so in terms of regulation and
it's not easier.
Yeah.
Oh. Like I think you have
something to think about. True, that's true. Yeah, no one would care, right? Those are very good points. All right, so we haven't heard from 1, 2, 3 plus. I'm going to answer the point in. We haven't heard from 4 of you yet.
I also disagree.
You disagree?
So I think there's like a and then what's not what's gonna happen in the future. And also like I believe like for every business that we have, it's like the liability is in the nature. So I kind of feel like say with nature that find the its probability. So I kind of feel like maybe we need to look more into the
That is also a very good point. So right now, again, this is under United Nations. So for anyone who may not be familiar with malaria, it was actually discovered from nature. So I think at that point it actually was learned from, I think it was from a tribe where people sort of realized, hey, like what you're doing works, let's actually convert this into, like a modern answer. So what group under United Nations is doing is developing West, where they are sort of looking at natural flora, natural plants across all the United Nations countries, and really just trying to encourage them to actually look more into natural resources. that might be helpful in this. Very interesting point. Thank you. All right. Who wants to go next?
I disagree.
You disagree? Okay.
Yeah. Although the African have many potential and provide personalized treatment and physical technology, and as the Asia and they have ongoing side effects. Agriculture traditional medicine is more serious.
Okay, all right. So Son's point was also very interesting. Even though this is sort of leading towards the idea of personalized medicine, which is what a lot of people are opting for right now, we still don't know what's going to happen. You are right. Thank you for that. All right, so I'm going to go Gao first and then Anisha, you go last. Gao, what do you think?
I think I think yes.
You think yes, you think it's good.
Yeah.
Would you edit the human or would you edit the microbe? Of course.
Human or microbe?
Yeah, so would you edit like TB itself or would you edit the human? So we heard sort of different opinions. There are some people who sort of say, well, if you edit the microbe, then it's not a problem because there's no ethical issues, but how do you deal with it? While there were others who sort of say, well, if you edit human and sort of change chemical changes to the immune system, then you don't have to worry about drug resistance.
Okay.
Which one? Human or microbe? Both. Or if you think you may not want to use it for TB, but you can think of some other situations that you might use it in. Or if you think all of this, nothing works. People just need to come up with a new treatment for TB.
Because all of new technique maybe not. It is have some disagree or somewhat, but in the future, it is all right.
Okay, so at the moment you disagree, but in the future you think this is something that you might be okay with because there will be more information about anything. Is that accurate to say that was what you think? Okay. We'll come back to Gao if you have anything to add. Thank you, Gao, so much for sharing your opinion. All right, Anisha, you're last. I'm just going to count you in as an unofficial member of this class.
I'm okay with editing the microbe, but not the human. I think even if everything is perfect, no side effects, nothing, the public won't accept it. Like even with the corona vaccine, there were so many conspiracy theories. So the government is planting microchips and all that. So there's going to be conspiracy theories with everything.
So how long, this is probably a question for everyone, how long, as scientists, how long do you think we should wait before a technology is acceptable? So most people receive the vaccines for COVID in 2020. It's now 2024. Do you think people are more open to COVID vaccines now? It's 4 years. You think yes? You think if like now if I tell everyone, guys, we have KP1, KP2 in the country, everyone needs another booster shot of COVID vaccines, who would actually be okay with that? Yeah, because there's a new variant right now, KP1 and KP2.
For me, the first it's okay, but the outcome of the vaccine didn't like show that much, sick, because when the parents came, everyone got again. So everyone was saying, okay, going on with the vaccine, that me too, because as you know, like this year we took it like when we were
like really young.
Yeah, when we were like just 1 year, I don't know, like till now like it's working. So
that's true. Okay.
Yeah, so right now I don't think so. Like, everyone don't care about the advancement of that vaccine that they do
versus whether it's efficient or not.
Yeah. The efficiency, I don't think so. Like,
okay.
About
that's a fair point.
Like as a community, but about science maybe something else, as we know. But I don't think so. Right now, people taking care about treatment. It just doesn't make any sense.
Okay, yeah, I think that's a good point, right? So if people can prove that it's efficient, just don't test it in me, go test it in someone else, and then if it works, then people might be more open to it. Maybe.
More open to COVID vaccine nowadays. Because when we see AstraZeneca vaccine, they take down all the products because there's an efficacy problem. There's also the safety which caused death due to its cost. So people will like, got new vaccine that I want.
So I think for all of you being scientists who may potentially be involved one day in the development of a particular product, there are so many aspects to think about, right? Is it something that I'm going to call it future proof, right? So is it something that once you develop, you don't have to worry about anymore? You pointed out BCG is awesome, right? So how do you actually ensure that you can develop something that works forever? A lot of people pointed out you don't know how safe it is, but if you don't test it on people, how would we know whether it's safe or not? So how many people should we test it on? Or is it safe to say, okay, I'm just going to test it on animals and then good, we can test it in humans? Or some people who are against like your computer, and you're like, no, how dare you treat animals that way? And you just want to sort of look at computer modeling. Would you say that it's sufficient to determine whether it's safe to be tested? So lots of different things to think about. So I think you guys are lucky because you're in a field where there's always something new. I think that's it for Hockey. So we ended a little bit early, but if anyone wants to add any last thoughts, any sort of opinions, you can. Otherwise, you can officially end for the week. Maybe I'll quickly go around the table. Now, anything you want to add or ask?
No.
No, you're good. Right, what about you? So, you?
No.
Okay. Unofficial number and no, Kusha? Yeah?
There's still like a few participants like.
Why would I?
Here.
That's a very interesting question. I don't know. So I think.
Oh, yeah, this question, our ideas, it was, it's not like, what's how, all this idea of
actually, so I think I
agree with this one.
Okay, so I think it's definitely, at least from what we've seen from our work, it's definitely easier editing the pathogen than human. So because we modified, okay, so just so that everyone know, what I do is I do genome editing as a potential gene therapy in HIV cure research. So we actually modify both the virus and actually the immune system. The virus is easy, you just knock out different genes, and we've sort of concurrently knocked out 6 different genes. The one that was tested in human, they knocked out 3. So we thought, okay, we tested in 6, that should be more successful. And the results were fantastic, but that's in some ways easy. because it's the pathogen itself. With the immune system, human immune system that we edited, that was a bit more challenging. So we edited that based on, there is a group of people known as elite controllers in the world. Elite controllers are very interesting because they get infected, but the viral load doesn't go up. So they sometimes have undetectable virus and their immune system is not affected. So with a lot of people living with HIV, usually their immune system sort of crashes, and that's why we end up getting AIDS. But in people who are elite controllers, that doesn't happen. So we tried mimicking their entire immune system in normal progressors, that is people who have gotten HIV before, just to see how it changes. We tested it across a lot of different cohorts. So we tested it in cohorts where people were just newly diagnosed. We tested it in cohorts where people were already on antiretroviral treatment. We tested it in young people, old people, male, female, anything and everything you can think of, the parameters we tested it in. But the results were really very different. So that we couldn't actually conclude, like there was nothing that we could sort of look at and sort of say, yes, like this is when and how the gene therapy should be done if you're trying to edit the immune system. Because as one of you pointed out earlier, it's complex. And that's why our immune system is the immune system. So me personally, I would say it's definitely worth trying out just because I think with TB, the multi-drug resistance that we see in TB is pretty bad. I think it is getting progressively worse over time. I don't really sort of see, at least right now with the current technology that we have.
Thank you.